Hi everybody. We are going to be learning about mitosis today, so let's go ahead and get started. So our learning objectives for today, we are going to be learning about the differences between sexual and asexual reproduction. We are going to be summarizing the steps in DNA replication. We are going to be learning to tell the difference between sister chromatids and homologous chromosomes, and we are going to learn how to explain the steps of mitosis and know what chromosome number we have in the cell at each step. So, mitosis is the process of cell division, and cell division needs to happen when an organism is growing, because the only way for an organism to grow is to create more cells. And then healing, things like wounds or sicknesses, that is another reason that cells will need to divide to replace damaged cells, and then to create new organisms. And here are some other examples to the right. So we have two different types of reproduction. We have sexual reproduction and we have asexual reproduction. Sexual reproduction requires mitosis and meiosis. Meiosis will be the next set of slides. This one is mitosis. So if we look here, we've got mitosis right here and here, and we have meiosis over here. So these two mitosis sections, those are an example of growth, like we talked about on the last slide. And over here, this is an example of new organism. Okay, so take a moment and answer this iClicker question. Okay, but now I'm going to discuss it, and so if you are a little bit confused, you can listen to the discussion and then take a moment to answer. Okay, so the offspring in this diagram is our little human baby right here, and this human baby is the child of these two parents. So we've got parent 1 and parent 2. So parent 1 is going to donate some genetic material in the form of a gamete, right? And parent 2 is going to donate genetic material in the form of a gamete, but neither one is going to give all of their genetic material, because we have to have the same number of chromosomes in this offspring, right? So each of these gametes has half of the normal number of chromosomes. Then when fertilization occurs, the gametes combine and we go back to our normal number of chromosomes. So now this fertilized zygote is 1/2 P1 and 1/2 P2. And this zygote is going to go through mitosis, cell division, and we are going to get our little human baby right here. So is the baby genetically identical to either of the parents, P1 or P2? Go ahead and put in your iClicker answer, and we'll move on. Okay, now an example of asexual reproduction. Remember the prefix A over here means not. Okay, so this is not sexual reproduction. An example of asexual reproduction is called binary fission. Binary meaning 2, and fission means like cutting or dividing. Okay, so this example is a bacterium. Our bacterium is going to duplicate their chromosomes. So we have one chromosome here and we have 2 chromosomes there, circular bacterial chromosomes. Then our cell is going to start expanding and pinching the cell membrane in the middle, and our chromosomes are going to go to either side. Okay, then that membrane is going to be recreated in the middle, and we end up with two cells down here. Okay, so remembering that our chromosome in cell 1 and our chromosome in cell 2 were copied, is the resulting cell, are these two cells genetically identical? Okay, Go ahead and answer that in iClicker. Okay, there is another form of asexual reproduction, and so the one we just discussed is on a cellular level, but asexual reproduction can also occur on an organismal level. So over here, of course, we have SpongeBob, and SpongeBob canonically can reproduce by budding. So we have our adult parent here, so this is our parent organism, and then we have all of these offspring, right? So SpongeBob has copied his DNA and passed it on to these little bud organisms, right? Another example on this side, this plant is called Mother of Thousands, and each of these little buds on the side of the leaf is going to be able to grow into an entirely new organism. So this parent plant, right, has copied its DNA and put it into these little offspring buds, and then those will be able to grow into new plants. So keeping in mind that our parent organisms are copying their DNA and budding off into new organisms, and there's no second parent organism here. Are these offspring genetically identical to the parent? Go ahead and answer that in iClicker. Okay. Another thing we need to talk about is cell death. So cell death is an important part of life as a whole. So programmed cell death is called apoptosis. Apoptosis is an intentional killing of a cell. An example of this is the development of digits in organisms. So we can see two examples here of an early development foot, right? So we can see our little digits, and then we can see we've got webbing here between the digits. Okay, so in our duck over here, we are not going to see any programmed cell death, right? So it's going to go through the rest of development, and we still have our digits, and we still have the webbing. Okay, but in a chicken foot, we have the digits, we have the webbing, but at some point in development, these cells in the middle are going to be signaled to die through apoptosis. So we'll do that in black here. Okay, and now notice we no longer have webbing between the digits of the chicken foot. Okay, so this one went through apoptosis, right? Now, in the cell cycle, we have several stages. We have the G stages. G stands for growth. We have the S stage, which stands for synthesis. And we have the M stage, which stands for mitosis. Okay? So in the growth stages, our cell is, it's going to be an interphase. It's going to be expanding its membrane, expanding its cytoplasm. In the S phase, we're going to start getting ready for our cell to divide. And we're going to do this by copying all of our genetic material. So synthesis is the step in which DNA replication occurs. Okay, and that means we're making a copy. All right, so looking at our DNA over on this side, we've got our cell, and in our cell we have our DNA in the nucleus, and we can see our double helix that we learned about in our DNA section, and we can see our hydrogen bonded nitrogenous bases along the middle. And now at the bottom of this image, we can see our replication bubble open. So now we have one strand that has its nitrogenous bases exposed on the left, and we have the other strand that has its nitrogenous bases exposed on the right. So now our enzymes can get in and they can start making a copy, a complementary DNA strand for each one of these. And so at the end, one strand is going to be made of old DNA and new DNA, and on the other strand, one side is going to be made of old DNA and the other is made of new DNA, okay? So our copied strands are half old, half new, so that they are exact copies. So just as a review from last time, go ahead and answer in iClicker which protein makes strands of DNA. Okay, we've got helicase, binding proteins, primase, DNA polymerase, and ligase. So which one of these makes our strands of DNA? Go ahead and answer that. I'm going to move on to the next slide. Okay, so now we can see all the roles of these proteins. The first one we see over here is helicase. Helicase is responsible for opening the replication bubble so that we can access each side of the DNA double helix. Binding proteins, we're not going to worry about too much. They're just helping the process. Primase is going to be adding an RNA sequence, and this tells polymerase where to go. Where to go, okay? And then we have DNA polymerase. DNA polymerase, remember, poly means many, and ase means it's an enzyme. So we're making a many nucleotide long strand of DNA. Okay, so our DNA polymerase is going to bind at this primer, right? Because primase has told DNA polymerase where to go. And then it is going to start adding nucleotides, and then we're going to have our new strand of DNA. And our last protein here is ligase. Ligase, also an enzyme. The four that we're talking about here are enzymes. Remember that ase equals enzyme. Okay. Ligase is going to be doing the cleanup. So it will be bonding fragments of DNA by forming covalent bonds between the phosphate sugar backbone, so that we have two whole strands of DNA. Okay. All right, so the next thing we need to know before we talk about DNA replication is that DNA replication, or DNA in general, has directionality. Okay, so, and what I mean by this is that our nucleotides, remember we have our sugar, we have our phosphate, and we have our nitrogenous base, right? And because every nucleotide is oriented in this way, we're going to end up with a direction, right? So this is one end, so this would be the 3' end, and this would be the 5' end. Oh my goodness, the 5' end. So every nitrogenous base, or every nucleotide here, is going to have a 3' end and a 5' end, which means that our DNA strands also have a 3' and 5' end. So you can see here's the 5' and here's the 3'. And on our complementary side, the 3' is at the bottom and the 5' is at the top. So the reason that we have these 5' and 3' ends has to do with carbon counting. So our sugar has these 5 carbons. We have carbon 1, 2, 3, where we have a bond, 4, and 5, where we have a bond. So at the 5' we're going to have a phosphate, and at the 3' we're going to have a phosphate. Okay? And so this makes up our phosphate sugar backbone. So on the 3' end, this 3' carbon is exposed to and available to bond. And on the 5' end, the 5' carbon is exposed and available to bond. okay? Now, because our complementary strands, their 5' and 3' run in opposite directions. So you can think of this as like a two-way traffic on a street, right? So our 5' and 3' on one strand complementary is going this way, and then our 5', 3' in the other strand is going that way. And this trait is called anti-parallel, okay? So they're both running in the same direction, but the traffic is going two directions along that line, okay? So let's go over some terminology real quick. Primers, remember that is a short sequence of RNA that tells DNA polymerase where to go. When I refer to 5' to 3', I'm referring to the direction that DNA polymerase moves. Okay, remember polymerase is adding nucleotides and it moves from 5' to 3'. The leading strand, this is going to be the strand that is able to be extended continuously. The lagging strand, remember our strands are anti-parallel, so while one can extend continuously, the other one gets replicated in fragments, and that's called the lagging strand, okay? And then those fragments on the lagging strand are called Okazaki fragments, okay? So this, I know, looks like a lot, but we're going to go through each step and each protein as it's involved. So in this first part, we have DNA helicase. DNA helicase is opening the replication bubble. So this section here, this is called our replication bubble, and helicase is moving this way. Okay, at this point, our RNA primers are going to be added. So that's the job of primase. So primase is here and here, and it is adding these short sections of RNA that are going to tell polymerase where to go. Next, we have polymerase. Polymerase is here and here. Okay, now this strand on the top, this is our leading strand. Leading strand. The new DNA that we are making here is going from 5' to 3'. So DNA polymerase can continue moving as helicase moves, right? Because helicase is moving in the same direction. On the other strand, we'll do this in a slightly different shade of green. On the other strand, our new DNA is anti-parallel. So the 5' end is towards helicase, and the 3' end is away from helicase. And polymerase can only go from 5 to 3. So in this set, polymerase is moving in the opposite direction of helicase. And so this is where we're going to see those Okazaki fragments. This is the lagging strand. Okay, and so you can see that up here in this one. We've got our leading and lagging listed. And you can see now what these fragments look like. We've got one fragment here and one fragment here, right? So leading strand is a solid continuous piece. Lagging strand has Okazaki fragments, okay? And then our last little step here is ligase. So ligase is this little almost rectangular one. And ligase is going to come in here and pretend this is our Okazaki fragments. It is going to create a bond between our Okazaki fragments so that our fragments become one continuous piece. Okay? Now, our DNA can be either tightly packed or loosely packed. So if we have loosely packed DNA, this is going to allow for expression. Remember, expression is when a gene can be transcribed and translated into a protein. Okay, so relaxed chromosomes lead to expression. tightly coiled, this is more for protection. Okay, when it's tightly coiled, the DNA is less likely to be damaged. And when we need to move all of our DNA from one cell to another cell, it's important that we not damage it. So tightly coiled means protection, and this is getting ready for cell division. Okay. Now, we're going to talk about the difference between homologous chromosomes and sister chromatids. So when DNA is tightly coiled, we call that chromatin. And so both of these words start with the chroma prefix because they are made of chromatin. So in this situation, I'm going to change the color of one of our chromosomes here because it was a little confusing for my other class. So we're going to make this one blue, okay? So these are two homologous chromosomes. Homologous means that they have the same genes, but different versions. Okay, so this would be like if there was a gene for eye color on the chromosome, maybe our purple chromosome is for blue eyes and our blue chromosome is for brown eyes, right? The same eye color gene, but different versions, okay? So in this example, our left chromosome is going to be our P1 from parent 1, and our right chromosome is going to be P2 from parent 2. Now, our chromosomes are going to get duplicated in preparation for mitosis, and these are still going to follow our pairing rules, okay? And this is replication, it's all DNA, so we have the T, right? So A pairs with T, C pairs with G. So now we are going to see a copy of P2, and it is going to be connected at the centromere. Okay, and then we are going to see a copy of P1, connected at the centromere. Okay, so P1's copy we're going to call P1A, and P2's copy we're going to call P2A. So if P1 was completely exactly copied, then P1A is going to be identical to P1. Okay, so P1A will be identical to P1. And P2 was exactly copied to make P2A. So P2 and P2A are going to be identical, right? So P1 and P1A identical, P2 and P2A identical, but P1 is not identical to P2, and P1A is not identical to P2A, okay? And for that matter, P1A is also not identical to P2. Okay, so homologous chromosomes are not identical. The copies that are connected at the centromere are identical, they are the same. Okay, so for this, I want you to answer the question of which chromatid is identical to the circled chromatid. Okay, and so I'm going to circle this one. Right, so I've circled that chromatid, and our chromatids are, we've got this one, this one, this one, and this one. Okay, so on your iClicker, go ahead and select which of those 4 chromatids is identical to the one that I just circled in blue. Okay. Okay, so let's work this out. I'm going to go over to, let's see, am I on this side now? Oh, there we go. Okay, I'm going to go over here to paint, and we're going to look at this again. We're going to look at this with some different colors. So we've got, we'll say that's one chromosome, and that's the other chromosome, right? Okay, so we're going to call this one P2, and this one P1. Okay, P1 is going to get copied. And it's going to be connected at the centromere. And then P2 is going to get copied and connected at the centromere. Okay, so I circled this chromatid right here, the yellow one. So we just said that the yellow chromatid is an exact copy of P1, right? So we would indicate P1 in our question. Now, P2 has an exact copy, but P2's exact copy is this chromatid on the right, okay? So these two are sisters. They are copies. And these two are sisters. And all of this is homologous. Okay? So let's go back over here. Hope you took a moment to answer our iClicker question, and we are going to go on. I do want to just make one more reminder. Actually, we'll move on. Okay, so now we're ready for mitosis, right? So we were previously in the S phase where we were replicating DNA. Now we are in the M phase where we are undergoing mitosis. Okay. So our goal in mitosis is to split up sister chromatids. So these, on this chromosome, when it has a centromere, when it is connected at the centromere, it is a chromosome. It is one chromosome, okay? Our goal in mitosis is to separate our copies and put one copy in one cell and put the other copy in the other cell, okay? So then we're going to end up with one chromosome here and one chromosome here. Once the chromatids are no longer connected at the centromere, they are counted as two separate chromosomes, okay? So this is a chromosome, and this is a chromosome. When they are connected at the centromere, also a chromosome, okay? So centromere is a chromosome, disconnected, two chromosomes, okay? Now, here are our stages of mitosis. I just wanted to show you the whole picture before we go into each step. Now, in our example cell has 4 chromosomes. If we want to end up with identical daughter cells, how many chromosomes are going to be in each daughter cell, okay? So here's our parent cell. We've got 1, 2, 3, 4 chromosomes. We want to have 2 exact copies in mitosis, okay? So tell me how many chromosomes are going to be in this cell if we're getting 2 exact copies. So we're going to go through it bit by bit. So prophase is first. prophase is preparing for division. In prophase, our chromosomes are going to condense, and our cytoskeleton is going to get ready to pull those chromosomes apart. There's a lot more about the cytoskeleton. There's terminology that we're not going to get into today. So just remember that in prophase, the cytoskeleton is getting ready. And the cytoskeleton is made up of fibers and proteins. Fibers and proteins. Okay, so we're going to count the chromosomes here, and you're going to answer how many chromosomes are in the cell during prophase. So I'm going to get a little bit of a brighter color. We're going to do orange. So we've got 1, 2, 3, and 4. Okay, so how many chromosomes do we have here? We have 4. All right. Next stage, we have prometaphase. This is a transitional step, and it's been called several things over time, over iterations of textbooks. Currently, it is called prometaphase. And in this stage, our nucleus is going to break down so that our chromosomes can move freely, and our cytoskeleton is going to connect to the centromeres of our chromosomes. So now we have these fibers and proteins that are connected to our chromosomes and are going to start moving them around. Okay. How many chromosomes do we have here? We have our X's, right? So when we have an X, that's 2 chromosomes attached to the centromere, 2 chromatids, oh my goodness, 2 chromatids attached to the centromere, and this is not letting me draw a circle, and this is 1 chromosome. Okay, attached to the centromere, 1 chromosome. So we have 1 X, 2 X's, 3 X's, and 4 X's. That's 4 chromosomes. Okay. Next, we have metaphase. Metaphase, you can remember, is when they're lined up in the middle. Okay. And in mitosis, our chromosomes are lining up single file, just one line of chromosomes. Okay. So here's our line of chromosomes. They are still X's, connected at the centromere. So go ahead and count those. Put your answer in the blank. In anaphase, we're being pulled apart. So anaphase, apart, another direction, whatever helps you remember. Okay, this is where our sister chromatids get separated. This is where the centromere is going to separate, and our 2 chromatids are going to be pulled apart. Okay, so remember, if it is an X connected at the centromere, that is 1 chromosome. If they are no longer connected at the centromere, each one is its own chromosome. So that would be 2 chromosomes. Okay, so we're going to count here. There's no centromere. We have 1, 2, 3, 4. On the other side, 5, 6, 7, 8. Okay. So how many are in this stage? In anaphase, we now have 8 chromosomes in the cell. Our last stage is telophase. You can remember this is the termination stage of mitosis. So in this stage, our cell's going to get ready to become 2 cells. So we can see our membrane is getting a little bit wider. Our nucleus is reforming. Our chromosomes are separated to either side. But this right now is still considered 1 cell. 1 cell. Okay, so our chromosomes relax, 2 nuclei are forming, but go ahead and count the chromosomes in this one cell and put your answer here. How many chromosomes are in the 1 cell? Okay, and then we have one more stage. One more stage, and I'm going to go ahead and clear this and show you that. And this stage is not in mitosis technically, but it is important to remember. So we go from telophase, where our cell is elongated, we go to cytokinesis, where we are going to have two cells form. Okay, so we've got our chromosomes there, and our chromosomes there. Cytokinesis is not a part of mitosis, but it is when our cell actually splits into two. Okay, so previously there was a connection here where we had cytoplasm. Now it is broken. Now we have two cells. One cell, two cells. At this stage, we go back to having 4 chromosomes per cell. Okay? So go ahead and finish up that iClicker question. Now, just to review, we're going to do like 2 questions on this slide. There is a question up here at the top. I worded it poorly, so I am not going to count it. Okay, so for this question on the iClicker, just pick any answer. I'm not going to count it. So next iClicker question is during metaphase, how many chromosomes are lined up in the middle? During metaphase, how many chromosomes are lined up in the middle? Okay. And then the next question here is during anaphase, how many chromosomes are being pulled apart? Okay. Sister chromatids have separated, centromere is no longer connecting them. How many chromosomes are being pulled apart in anaphase? Go ahead and answer those. And I'm going to run through one practice, but for the sake of time, we're not going to go through the other practice. So just to start with, actually, you can work on this practice on your own, and if you have any questions, you can email me because this is a very long video already. So we've gone through the stages of mitosis, we've gone through DNA replication, and there are 2 practice scenarios for you to work on at the end here. And that is it. I'll see you next time.